Yeah, got it. It's 10 minutes past 12 in Ankara, 10 minutes past 10 in Nigeria. Thank you for joining us. A warm welcome to everyone. Thank you for joining the quarterly PSI webinar series, today being the 29th of January, 2023. My name is Alkasim Hassan, and I will be the moderator for today's program. Today's program will feature an invited guest, but before then, let me briefly take you through some announcements. Please note that this webinar is being recorded, and the recording will be available on our website. Feel free to ask any questions by using the chat box. And you may provide feedback via email by sending email to pharmscholarinitiative@gmail.com. It's my slide changing? Oh, okay.
It's not
for the benefit of those joining us for the first time. Just a minute. Yeah, let me take you through what PSI stands for, or what we stand for at PSI. Sorry, PSI is simply means Pharmacy Scholar Initiative. It's a mentorship network for pharmacists who have keen interest in research and career development. This group was founded in December 2017, and it is registered with the Corporate Affairs Commission. Membership cut across different parts of the world. Our objectives include to assist young researchers in the formulation of research ideas, design, conducting, and reporting this research, to support young researchers on scientific writing skills that is required in writing proposal, thesis, as well as manuscript. We also share learning materials, and we provide guidance to young scholars on career development. Like I mentioned, today's event is the first quarter webinar series, and we have our invited guest already joining us. So based on today's agenda, we have welcome address by the PSI President, Dr. Ibrahim Jato, introduction of our guests, then the presentation proper, and Q&A, followed by closing remark and presentation of certificates by the Academic Planning Committee. So without further ado, I will now invite the PSI President, Dr. Ibrahim Jato, to give his welcome address. Thank you.
Assalamu alaikum wa rahmatullah. Because our guest speaker doesn't have much time, he has only 1 hour, and we've already gone like 15 minutes out of the time. So there is no need to waste so much of your time. Assalamu alaikum. My name is Ibrahim, and I'm the one coordinating the activities of Pharmacy Scholars Initiative. I would like to use this opportunity to welcome everyone to this webinar, and it is my belief that we all learn something new out of this webinar. As you can see, the title of the presentation is very catchy. So, and I'm very sure all of us would like to hear or would like to see how microorganisms whispers in their own world. Thank you so much and enjoy listening. Thank you.
Thank you. That was a brief welcome address by the PSI President. I will now invite Amira Abdullahi to introduce our guest speaker for the day. Amira, over to you.
Amir, are you there?
Assalamu alaikum.
Wa alaikum assalam wa rahmatullah.
Wa alaikum assalam wa rahmatullah.
Assalamu alaikum. Good morning, everyone.
Yes, we are hearing.
Okay, I'm here to introduce our guest speaker for today, Pharm Abubakar A, Falaki. Pharm Falaki is a graduate of Ahmadu Bello University, Zaria, where he did B.Pharm and MSc Pharmaceutical Microbiology. Our guest is currently a PhD candidate at the University of KwaZulu-Natal, South Africa, where he is working on the interspecies interaction of endophytic bacteria. Pharm Falaki is a staff of the Department of Pharmaceutical Microbiology and Biotechnology, ABU Zaria. He has delivered lectures and practical courses on pharmaceutical sciences. He has also participated in many training, workshops, and conferences worldwide. Falaki has many scientific publications to his name. His current research focuses on screening the inhibitory effect of compounds from endophytic bacteria on various quorum sensing systems and investigating the interspecies interaction of these endophytes using a high throughput binary approach. Pharm Falaki is happily married with children. Please join me in welcoming our guest speaker, Pharm Abubakar Ahmad Falaki. Thank you.
Thank you so much.
Thank you, Amira, for that brief biography of our guest speaker. So I will now invite our guest speaker Pharm Abubakar Falaki to deliver his lecture on titled The Whispering That Occurs Within the Invisible World of Microorganisms. You have the floor, sir.
Right. Okay, thank you very much, Amira, for that generous introduction. Good morning, good afternoon, or good evening, wherever you're connecting from. Thank you for inviting me also for this presentation. Brahim, am I
Yes,
loud enough?
yes. Yeah, audible.
Yes,
Yeah,
all right.
You're audible. Please go ahead.
All right, thank you very much. So we all know that microbes are said to be the oldest living organisms on Earth, and they've been around for not just millions but billions of years. And we all have this amazing interaction with these little creatures. And we also have like 10 times more bacterial cells than we have human beings, human cells on human beings. And this is the main reason why it is important to study microorganisms. Now, they are so important to us because in a unique way, they do keep us alive. They keep the environmental assaults out so that we can stay healthy. For example, they help to digest our food. They educate our immune system to get bad microbes out. So they do all these amazing things to us. Now, they also do a lot of terrible things as well. Now, we have a bunch of notorious microorganisms that if present in or on us, they make us really, really sick. Now, the question is, how are these tiny creatures able to do these wonderful tasks while being incredibly small? Now, that brings us to the topic of today. And Ibrahim Joto, I'm using the word whisper, given how tiny these microbes are. Now, it has been discovered that microorganisms are able to form groups, or what is called known as a quorum. I'm trying to move this. Okay. So this usually refers to the minimum amount of members in an organization, such as a legislative body needed to conduct business. Now, how can bacteria, this primitive little tiny organism, tell the difference from the time they are alone and times when they are in a community or in a group? And then they do all these things together. So it was discovered that they do this by talking or speaking to each other using a chemical language known as quorum sensing. Now, quorum sensing is just the regulation of gene expression to fluctuations in the cell population density. Now, the first such system was described in Vibrio fischeri, which has a special property that it makes light using quorum sensing system. Now, this is a symbiotic species that provides its marine host, which is the quid, with light, and this helps it to prevent predators from eating it. Now, the diagram on the right shows cycle. You can see a cycle there being your bacterial cells, and when it's alone, it doesn't make any light. But what it does is it produces and secretes molecules that are known as autoinducers, which are the triangles by the right-hand side. And when this bacteria is alone, the molecules just fluctuate around it, so there won't be any light. But when the bacteria now increases in number, they all participate in making these molecules, and thus they increase in proportion to their number. And when the molecules get to a particular amount and tells the bacteria how many there are, they recognize that molecule, and all of them now, or all of the bacteria now turn on the light in synchrony. Okay, right. So the molecules these microbes use to communicate are called autoinducers. And just like we have various different languages, these microbes also have their own different languages. For example, the gram-negative bacteria, they use the N-acyl homoserine lactone. For example, the Chronobacterium and the Pseudomonas. The gram-positive, such as Staphylococcus and Enterococcus, they use the oligopeptides. And then we have a universal language in which both gram-negative and gram-positive microorganisms use, and it's called the autoinducer tool. Right. So by attenuating or disrupting this quorum sensing, which is called quorum quenching, this disarms the virulence factor rather than killing the microorganism. Now, like I said, these microbes use the autoinducer to communicate, right? What is important is that the bacteria have a lot of behaviors that they carry out in this collective fashion. And this, by the right-hand side, includes virulence, bioinformation, bioluminescence, which is production of light, and motility and clumping. So you have all these amazing activities that they carry out when they're in a group, right? Now, this slide here shows examples of some of the experiments that are carried out in our lab. Now, the first example here shows inhibition of bioluminescence, which is a quorum sensing controlled phenotypic characteristics by Vibrio Havi. Now, we use extracts that were obtained from endophytes, which are microorganisms that grow within the internal tissues of plants. Now, we use this now to show how the autoinducer system actually works. So this is an agar well diffusion assay that demonstrated the inhibition of bioluminescence without any visible effect on the bacterial growth, because that's what you don't want. So now the figure on the right-hand side shows Chronobacterium violacum, which is a gram-negative bacterium that produces the characteristic purple pigment, which is called violacin. And this is mediated by what is called the acyl homoserine lactone quorum sensing system. Now, I deliberately brought this plate to show you the example of, or the difference between quorum sensing inhibition and the normal killing of bacteria. You can see the inner zone here shows how the killing happens, and the outer zone is what is the quorum sensing inhibition, which means it's inhibiting the violacin that produces the purple color without actually killing the microorganism, right? Now, apart from this, we have the quantitative quorum sensing inhibition assays that are carried out to determine or to confirm whether the inhibition of your violacin or bioluminescence is primarily due to killing and not due to killing, primarily due to killing and not quorum sensing inhibition, right? Now, like I mentioned earlier, one of the things that the microorganisms do is biofilm formation. And this shows a diagram of how the biofilm is being formed. The biofilms are just structural communities of the bacterial cells that are formed through quorum sensing, and they become enclosed in a self-produced polymeric matrix, right? So they are sort of protective mode of growth that allows the microbes to survive in hostile environments that are highly resistant to killing. And these are found mostly everywhere in nature, including rivers, pipes, and of course our human body. Now these biofilms often cause infections that are associated with medicinal implants, such as the catheters and other medical devices. Now, this is the main reason why you find that it's quite difficult to treat infections that are caused by these implants, especially for those that are using the intrauterine devices and other heart implants and so on. So in summary, because I made these slides to be as short as possible because of time. So in summary, what we discuss is how these bacteria are able to talk to each other using chemical words, and then they can carry out these tasks that we simply cannot accomplish if they act alone. And they can distinguish themselves from others by using these molecules that we call the autoreducers. And then finally, this is, it has been practically shown that having anti-quorum sensing agents developed as kinds of therapeutics takes care of the problem of the resistance that we're always having. Now, I talked about biofilm quite briefly because there's no time, but if I'm able to have more time in the future, I'll be able to discuss how these biofilms are being formed and the targets in which you can always find agents that will either prevent the biofilm from forming or disrupt them while they're at the final stage. So this is just a brief discourse of what I'm going to present. It's not too long. And thank you for your time. And if you have any questions, I'll be very happy to take them.
Okay, thank you very much, Pharmacist Abubakar Falaki, for the wonderful and brief presentation. I will now invite the participants to forward any questions or comments you have regarding the presentation. You can forward your questions via the chat box, or if you want to speak, you can unmute yourself and ask your questions.
Can I ask a question?
Okay, go ahead.
Yeah, thank you so much, Pharmacist Falaki, for this very beautiful and educative presentation. It might be possible I missed the information, or I'm just trying to ask, this signaling or the communication between and this bacteria, is this like species specific, or they can communicate to like intra-species communication? Like maybe a particular bacteria can communicate with another bacteria not in the same species. Like communication, is it interspecies communication, or it could be both ways, intra and inter?
Can I take the question now, or should I take all the questions at once?
Yes, I think you can take it since people are
Yeah,
still.
you can take it as they come. Since for now,
Right.
it's just a question from Dr. Jato.
Okay. All right, that's a good question, actually. I wasn't into details with my presentation because of time, but if you remember, I mentioned that you have a universal language that these microorganisms use to communicate. And of course, these microorganisms live in a complex community in which they interact with each other, right? And so they use these chemical signals, which are unique and can be either for intra or interspecies interaction, right? So imagine if I should use this analogy. If you go to a wedding, right? You do know that in that wedding, you will have all your relatives there, okay? So you're able to communicate with those relatives using your own peculiar language. But then you have people also that you don't speak the same language with. Maybe someone speaks Yoruba and you're speaking. So you can use English language now to speak to them. That's where the universal language comes in. So they do have a way of communicating based on what they want to achieve, right? That's why they are very clever little creatures, okay? So they do have communication between their own species, and they can also interact with other species to carry out whatever their task they intend to do together. I hope that answers your question.
Yes, sir. Thank you so much, sir.
Once again, comments, questions are welcome via the chat box or by unmuting yourself and asking the questions. Thank you.
Where are the pharmacology people?
It seems the house is quiet.
Dr. Mustafa from Malaysia, any comments? Wa alaikum assalam.
Yeah, I think he has done justice to the some of these things are really quite new.
Pharm Mustafa, can you please increase the audio?
We cannot hear you well.
We can barely hear you.
Maybe because I'm in a bus traveling from one state to another. So I just want to appreciate Dr. Falaki for the presentation.
All
Thank
right.
you very much.
Thank you so much.
Thank you very much.
Thank you, Mohammed. But
Mustafa.
don't allow my supervisor to hear you calling me doctor yet. extra weeks for me, please.
You know, at PSI, everyone is a doctor.
That's why you call you doctor.
Once you finish your B Pharm and you're either doing master's or PhD, you're on course to become a doctor. So we address you as doctor.
Yeah, I can see Abdurrahman Abdullah from National Eye Center.
Okay.
Abdurrahman. Okay. So anybody with comments? Abubakar Isa, Awal Bala, something Fatima Alijos.
At PSI, do you have microbiologists? Pharmaceutical microbiologists?
No.
I suppose no.
Yeah, we don't have. Baraka.
But those in pharmacology can relate.
Yeah. Anyway, I think we can, since I don't have much time.
So we now move to the next agenda, which is closing remark by the PSI Academic Planning Committee. So on this note, I will call on Dr. Awal.
No, you can call Zayan.
Okay, Zayan is in the house. Okay, I will call on Dr. Zayan to give a closing remark. Dr. Zayan?
Thank you so much, Dr. Alkasim. Don't let my supervisor hear you calling me Dr. Zayan. Well, it's a pleasure having you all, and thank you so much, Dr. Falaki, for the wonderful presentation. I think it was so concise that everybody understood where you are going to, and for acting on time and taking us through this interesting topic. We thank you so much for accepting the invitation and putting us through this. And also I have to thank the moderator for being timely and making it interesting. And I also have to thank all the participants for taking time to attend and also learn. I think it's been a wonderful session. Thank you so much, all. And for the presenter, we hope to have you again in the near future for a broader discussion. Thank you so much.
Thank you, Dr. Zayan, for the closing remark. So it's time for us to award our guest speaker certificate. So I will now call on.
Yeah,
Dr.
I don't know, Dr. Kankia, who is presenting this?
Basira, yeah, Dr. Basira Kankia, are you there?
Who's giving the certificate?
Dr. Basira Kankia? Okay, perhaps she's not available. Chato, who do you call on? It's Awal Bala available.
Sorry, Assalamu alaikum.
Sorry,
the certificates will be sent to the emails, inshallah.
Okay, I
I'm unable to share.
Can you see this? Can you this one?
Share the screen.
Can you see this one?
Yes, we can see it.
Is it the one by Falaki?
No, it's
okay.
It's not.
Sorry. Yeah, let me start with
the next one.
Kankia, you can read this. You can read the certificate.
Okay. This one for the moderator. So thank you very much. Okay. Okay, the presenters. Thank you very much, Dr. Abubakar Falaki, for the wonderful and brief presentation. We look forward to learning more on the topic, inshallah, in the near future.
All right, then this.
Thank you.
All right, then this.
Okay, thank you very much, Dr. Alkasim Hassan, for the wonderful moderation. And thank you for sparing your time to moderate the session. Thank you very much.
All right.
Thank you very much.
I think somebody is raising his hand, is it?
For now, it's none. It's just a message of appreciation from the participants.
Okay.
So if I cannot see any question in the chat box.
Okay. Dr. Falaki raised his hand. That's what my... Dr. Falaki, you want to say something?
Hello, yes, Ibrahim, I just wanted to add that actually the presentation had two parts. I wanted to talk about the quorum sensing and the biofilm part. Because I only had 1 hour, that's why I took out the biofilm part. So in case in the near future, if there's any need for me to come and talk about the biofilms in details, because it's actually a broad area, I would be really glad to share with you guys.
Okay. That would be fantastic. Yes, inshallah. Let me note it. And then inshallah, we'll surely invite you once again to present in the next part.
Thank you very much, sir. So on behalf of PSI, and all the participants. We've come to the end of our presentation, and we wish everyone a happy weekend and success in our future endeavors. Thank you very much.
All right. Thank you so much. Thank you, everyone, for attending.
Thank you.
Yeah.
Thank you.
Looking forward to see you in our next webinar. Thank you so much.